Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. And since the best way to support truly groundbreaking research coverage is, well, to spread the word, please take a moment to like, subscribe, and share this deep dive with your friends and family. It really supports the Sand Science Experience. Today, we're embarking on a deep dive into an enormously significant study concerning Alzheimer's disease, or AD. For decades, the therapeutic pipeline has really focused on clearing plaques, kind of the symptom, but this study uses a sophisticated approach called multiscale proteomic modeling, and the idea is to fundamentally shift the spotlight onto the functional components, the proteins.
That's absolutely right. Our mission today is to walk you through how this research moves beyond just, listing changes in the brain. They basically built an entire map of the functional protein networks. The goal, to identify key driver proteins, the actual causal regulators. And this offers a completely new list, really, of viable therapeutic targets for late-onset AD.
Right. We should probably start by just acknowledging the current situation with AD. I mean, it's the most common dementia.
Yep.
And while recent treatments like lecanemab and aducanumab have shown some, let's say, modest efficacy in targeting beta amyloid.
The classic target.
Exactly.
Yep.
We still have this massive knowledge gap about the underlying molecular mechanisms, especially for that sporadic late-onset form that affects, well, most patients.
Precisely. And, if we want to understand what's actually driving the disease, genetics only gets us part of the way there. It's just not the whole picture. That's why the authors pivoted to proteomics. Think of the cell less like a computer with code, DNA or RNA, and more like a busy factory. The proteins are the actual machines doing the work. So alterations in those protein machines, they're much more directly linked to the physical disease outcome, the phenotype, than maybe the changes you'd see in the blueprint.
Okay.
If the machine's broken, you need to analyze the machine itself, right?
Okay, so that's the why, focusing on the functional level, the proteins. Let's talk about the where and how, this multiscale proteomic modeling.
Yeah.
What does that actually involve? It sounds pretty complex.
It is. It's definitely a multi-step integrated approach. The multiscale part refers to integrating data from the highest level genetics all the way down to specific protein expression in key tissues, and then modeling how those proteins functionally interact. And they didn't just look at the whole brain either. They specifically targeted the Parahippocampal gyrus, or PHG.
The PHG, right? The memory center. That region is critical because it's one of the most susceptible areas to early AD pathology, isn't it?
Exactly. So they analyzed integrated proteomic and genetic data from nearly 200 postmortem brains. This was from the Mount Sinai Brain Bank. They compared healthy individuals to those with severe AD pathology, focusing right there in the PHG.
And the scale of the protein changes. It's just astounding. They found over 1,000 differentially expressed proteins, DEPs they call them. But what really jumped out at me is that over 70% of these were actually upregulated in the severe AD cases.
Yeah.
It's not just a system slowly winding down.
Yeah.
It seems like it's a system actively misfiring or something.
It really suggests an active, maybe even toxic response, not just passive decline. So to make sense of those 1,000 plus proteins, they moved into the second stage of their multiscale model, network analysis. They used protein co-expression tools like MEGENA to find these highly correlated clusters of proteins. You can think of them as functional teams. Then, crucially, they layered on Bayesian causal networks.
Okay, wait, let's stop there for a sec. What's the difference between those two steps? Co-expression and then Bayesian causal networks. Why do you need both?
That's a great question. So the co-expression analysis, MEGENA, it's kind of like drawing a map of which proteins are often seen together, at the scene of the crime. It identifies these modules based purely on correlation. But the Bayesian causal network step, that's the real innovation here. It uses mathematical inference to try and figure out the direction of influence. It moves beyond just correlation to infer which proteins might be the actual upstream regulators, sort of the causal puppet masters driving the expression of the others in that module.
Got it. So we've gone from a list of like 1,000 suspects to a map of how they interact and finally to a list of the inferred leaders, the drivers. And this network mapping, it immediately highlighted a really central functional cluster, didn't it?
It did. The analysis pointed directly to this key AD-associated subnetwork, which they named Module M3. And what's critically fascinating about M3 is that it seems to capture this thing we've suspected for a long time, but has been hard to pinpoint functionally, the toxic glia-neuron interaction.
Right, this module M3. It sounds like it visualizes this kind of cell-level civil war happening in the diseased brain. So tell us about the two sort of warring factions captured within M3.
Well, on one side, you have what you could call the victims, proteins associated with neuronal functions and synapse functions, and these proteins were uniformly downregulated. That reflects the failure of communication, the physical loss of neural connections, basically the foundation of cognitive decline.
Okay, so the functional machinery of the neurons themselves is just shutting down.
Exactly. But simultaneously, M3 captured the, let's call them activated perpetrators, proteins enriched in microglia and astrocytes. Those are the main glial support cells. And these were heavily upregulated. M3 specifically modeled the interaction where these activated upregulated glial proteins were functionally linked to those declining neuronal proteins.
Wow.
So it really strongly supports the hypothesis that dysregulated chronic activation and communication failure between the support cells and the neurons is a central driver of AD pathology captured right there in the proteome.
That structural map is fantastic, but we still have a huge target list. You mentioned the causal network analysis identified 580 key driver proteins, KDPs. I mean, if we're talking therapeutics, you can't target 580 things at once. How did the authors narrow that down? How did they decide which KDP really deserved the spotlight?
Yeah, that's always the challenge, the selection. They looked for KDPs that were, one, highly upregulated in AD, two, strongly connected within that critical M3 module, and three, their expression trajectory kind of mirrored the progressive genetic risk profile of AD, and this process led them directly to a protein called AHNAK, which actually ranked number 5 overall in their KDP list.
And you're saying this protein was previously, minimally studied in AD. Now it's a top candidate.
Pretty much. It hadn't received a lot of attention in the AD context before this, but their data showed AHNAK is consistently and significantly upregulated in AD patients. It's strongly associated with both pathological traits, like, say, protein accumulation, and also with the loss of cognitive function. And crucially, AHNAK's primary function seems to be localized in the astrocytes, placing it right in the operational center of that pathological Glia-neuron module M3. It's like a hub gene right where the breakdown is happening.
And the link to the biggest genetic risk factor, APOE, that really seals its significance, doesn't it?
Absolutely. They demonstrated that AHNAK expression wasn't just static, it progressively increased. It went up from healthy controls who had the standard APOE33 genotype to 80 patients who also had APOE33. But the spike was greatest in the highest genetic risk group, the APOE4 AID patients.
Okay.
So this data really suggests AHNAK might be a kind of molecular bridge where carrying the APOE4 risk genotype translates into active destructive disease pathology at the functional protein level.
That's incredibly compelling modeling and correlation data.
Yeah.
But the ultimate test is always functional validation. If you target AHNAK, does the pathology actually get better?
Yeah.
This is where they brought in the wet lab experiments, right?
Exactly. This is where the rubber meets the road. The researchers moved into their most demanding experiment. Using human-induced pluripotent stem cell models, iPSCs, they derived astrocytes from an APOE44 carrier that's the highest genetic risk profile. Then they co-cultured those human astrocytes with primary cortical neurons taken from a powerful AD mouse model, the 5x FAD mice. The question was pretty simple but profound. Can we reverse the disease phenotype in the neurons just by manipulating this one astrocytic protein, AHNAK?
Okay, so what were the first results, just looking at the astrocyte culture itself?
Well, knocking down AHNAK, basically silencing the gene in those high-risk astrocytes, immediately did something promising. It reduced two classic AD hallmarks, phosphorylated tau, or pTau, and beta amyloid levels right there in the culture medium.
Wow.
That's a huge early indicator that AHNAK is directly involved in producing or somehow regulating these toxic species.
But the real functional outcome, the proof, is in the neurons.
Mm.
What happened when they looked at the electrical activity in that co-culture system?
Right. And this is really the punchline that validates the whole network analysis approach. Downregulating AHNAK in the astrocytes led to a statistically significant, massive improvement in neuronal function. They saw a 1.8-fold increase in the number of spikes. That means better spiking activity, better bursting patterns, better overall network synchrony.
The 1.8-fold increase.
Yeah.
That's definitive functional evidence. Silencing this one astrocytic protein rescued the failing electrical activity of the neurons next to them.
It is. It's a really incredible transformation. It takes that abstract network concept and turns it into something measurable, a viable therapeutic strategy that literally improves brain function, at least in a dish model. And this wasn't just, a lucky strike. The validation loop closed perfectly. When they looked at which other proteins were changed by the AHNAK knockdown, that new protein signature was highly enriched right in the predicted network neighborhood of AHNAK from their original model. It confirmed the power of the initial multiscale modeling approach.
Okay, so if we take a step back and try to synthesize this whole deep dive, what's the core new understanding this research gives us about maybe the future of AD treatment?
I think the core understanding is really a shift in thinking about causality. Maybe AD doesn't primarily start with the neuron failing and accumulating plaques. Perhaps it starts with the glia, the support system, the astrocytes and microglia becoming chronically activated and actively failing to support the neurons properly. And AHNAK looks like a key operational bottleneck in that specific failure within astrocytes. So the researchers have systematically generated this list of hundreds of new potential targets, like AHNAK, targets that directly tackle this gliacentric failure rather than just, cleaning up the plaques afterwards.
That leaves us with a pretty provocative thought then. If the current approach often focuses on removing the debris left by the fire, this new approach seems to target the faulty electrical wiring, maybe, that started the fire in the first place. That feels like a fundamentally more impactful way to think about intervention down the line.
Indeed. I think we now have a much richer blueprint for the next generation of potential AD therapeutic targets.
Thank you for joining us for this deep dive into systems biology and potentially the future of Alzheimer's treatment. If you enjoyed hearing about AHNAK and the Glia-neuron subnetwork, please remember to like, subscribe, and share. We really look forward to having you back next time for another Sand Science experience.